Hi everyone, and welcome to our lecture on section 3 of chapter 16. I know I said in my remote learning welcome letter that we would be going from chapter 15 to chapter 32, but in the days of being locked in my house, I've decided to call an audible. We're going to do section 3 of chapter 16 before we go to chapter 32. And we would have done this if we were in class together, because this particular section of this chapter is about cancer. And not only is it about cancer, but it's about how the gene regulation systems that we've talked about ultimately fail or go wrong, and how we get cancer cells from those systems not working. So when we talked about, acetylation, methylation, transcription, translation, all those things, it's faults in those systems that lead or can contribute to cancer cells. So we're going to go from that stuff to this stuff. I have a blank version of the notes posted on Google Classroom, just like I would if we were in school together, but you by no means have to print it out. That's a lot of paper, and quite frankly, I don't know if you have the means to be printing out all that stuff like we would if we were in school. It's there for you if you want it, but I would encourage you just to follow along and listen. Maybe jot down some notes of your own on like a blank sheet of paper. It's totally up to you. All right, so we're going again from chapter 15 to chapter 16, and we're going to cover how the gene regulation systems go wrong and how that gives rise to cancer. So you should recognize some of the bolded words on the screen here: proto-oncogenes and oncogenes. So in chapter 9, we did talk about cancer. Chapter 9 was about the cell cycle. So we talked about how the cell cycle can go out of control. You remember our day where we talked about Henrietta Lacks? Remember that? One of my favorite days in AP Bio. But proto-oncogenes, do you remember what they are? Well, if you read the screen, right, or if you remembered, proto-oncogenes are the genes that deal in accelerating or causing cell growth to happen, essentially. They deal in stimulating the cell cycle. It's kind of like the gas pedal in your car, makes your car go, right? But things in the environment, like radiation, UV rays, things of that nature, they can mutate these proto-oncogenes. And when they become mutated, they become oncogenes. And oncogenes are the genes that cause cancer. They're like, imagine the brake pedal in your car being stuck down to the floor. That's like an oncogene. So the oncogene, remember, is a mutated version of the proto-oncogene. So how does that happen exactly? I just mentioned that things in the environment can cause it to happen. But there are 3 major ways that your book highlights in terms of how a proto-oncogene can turn into an oncogene. So the first one is here on this left-hand side of the screen. This is a translocation mutation, and you should remember what translocation is. Do you remember what it is? Yes, right? So it is the movement of a piece of DNA or a chromosome to another chromosome where it doesn't belong. Remember the Philadelphia chromosome, chromosomes 22 and number 9, remember that? Yes? Okay, so this is a situation where a gene moves to a new position, and when that happens, it can be under new controls. And if those new controls are doing a lot of transcription, a lot of translation, you're getting an excess amount of protein. And if those proteins are involved in the cell cycle, now you've got cell cycle out of control. Gene amplification, this is what the name suggests. This is where you have many copies of a gene, so it's kind of like duplication, where you now have extra copies of this one gene. So if you have more copies of it, that means transcription and translation would happen more. You would get more protein. And again, if this is protein that's involved in the cell cycle, now you've got out of control cell growth, which is ultimately what cancer is. We talked about tumors when we were covering chapter 9, so that's what's going on here. And then finally, a point mutation. You guys know what those are because we just talked about those in chapter 14, right? So that's your substitution, insertion, deletion. If you get a mutation like that in the gene, so at the DNA level, that can lead to an excess production of the proteins or the gene itself. We know that a change in the DNA ultimately is going to lead to a change in the amino acid sequence, which is going to lead to a change in how the protein is going to fold, and it's going to affect its ability to function. So these those three pictures and what's going on in those three pictures are outlined on this slide here. So these are the genetic changes that convert a proto-oncogene to an oncogene, and they fall into these three main categories that, again, go with these pictures here. So you have movement of the DNA within genomes, amplification, that's when you have multiple versions of the gene, and point mutations. This can happen in a control element or in the gene itself, and that can cause overproduction of a gene or an incorrect version of the protein, something like that. You should also recall tumor suppressor genes. So tumor suppressor genes are they're the ones that deal in breaking the cell cycle or causing it to slow down or inhibiting cell growth. This is like the brake pedal in your car. If proto-oncogenes are the accelerator, this is the brake pedal. And when these genes become mutated, now you have out of control cell growth. That's like when you go to hit the brakes and nothing happens. That would be a mutated version of these genes. But these genes actually do a couple other things. Not only do they help to control and promote normal cell division, they also deal in repairing damaged DNA, and we're going to look at a specific gene that deals in that, and you might remember it from chapter 9. They can also control that cell adhesion. Do you guys remember anchorage dependence and density dependent inhibition? Remember that picture in the book of all the cells stacked up on top of each other in the petri dish? So these cells help to deal in that as well. And then, of course, inhibiting the cell cycle, like I said, they are like the brake pedal for the cell cycle. All right. So we're going to look at a couple specific genes here that deal in cancer, and actually a lot of cancers, which is why we're going to look at them. So the proteins encoded by many proto-oncogenes and tumor suppressor genes are components of cell signaling pathways. And depending on when we're back in school, we're going to cover cell communication and cell signaling, or we may cover it remotely depending on how long this hiatus from school takes place here. But anyway, we're going to focus on two key genes, the RAS gene, which is a proto-oncogene, and the P53 gene, which is a tumor suppressor gene. Certain mutations in the RAS gene can impact cell division because the RAS cell signaling pathway is responsible for making proteins that stimulate the cell cycle. If there is a mutation in it, you have an excess in cell division. And I want to show you a quick video from the American Cancer Society about how this gene functions, because this gene is so important in lots of types of different kinds of cancer.
RAS is essentially the way that cells are constantly monitoring what's happening in the environment outside of them. And they're trying to make decisions as to whether they should divide, die, arrest, and not divide. The mutations that we see in cancer essentially leave the RAS protein switched on, but it can no longer be properly switched off. The easy way of thinking about it is that a cell is being told to grow all the time, even though there's nothing outside in the extracellular environment telling the cell to grow. If you take the most common of the RAS genes, which is KRAS, it's mutated in roughly a quarter of all cancers, for example, in 90% of pancreas cancer. It turns out to be a very difficult problem to make a drug to. I would venture to say that there will be a drug approved for a class of tumors with RAS gene mutations sometime in the next decade, maybe in 5 years.
Okay. So, whoops. Sorry. Okay. So that was a really great animation to give you an idea of what's going on with that gene. But here's a picture of what's going on. So the top is showing you a normal situation and the bottom is showing you the mutation. So in a normal pathway, so this is a cell signaling pathway. Again, we're going to get into the details of cell communication either remotely or hopefully when we're back in school. But the pathway is triggered by a growth factor that binds to a receptor that's in the cell membrane. So imagine that this area here is the cell and the cell membrane, and this is a protein that's embedded in the cell membrane. This would be, I don't know if you guys remember seeing the word ligand when we were talking about operons, but this is a little growth factor or a signaling molecule. And when that binds to the receptor protein, this signal is relayed by the RAS protein along a protein kinase cascade, they call it. So this is just a series of proteins, kind of like dominoes, that are passing along this message that has been received by the outside of the cell. And a transcription factor will be activated to make a protein to stimulate the cell cycle. So it's this kind of sequence of events here that it's the signal from the outside of the cell, it's the message getting passed along this protein kinase cascade, and again, we're going to get into the details of this in hopefully the next couple weeks here. That triggers the transcription factor to turn on and activate transcription, translation, protein. All right, that's the normal version, kind of like what you just saw in that video. The mutated version of that story, if you have a mutated RAS gene, it is abnormally active and results in excessive cell division, which can lead to cancer. And the gentleman in the video mentioned that it's about a quarter. Your book says 30% of human cancers, but essentially you have these cells that are, these proteins that are being made in excess when they're not needed. Even they're being made even though they didn't get the signal from the environment that says make more of these proteins. So they're just kind of going rogue and making lots of copies of these proteins. So after the RAS gene, one that's involved in a lot of types of cancer, the other one we're going to talk about is the p53 gene. And we talked about this one this year, way back when we talked about Henrietta Lacks. So you might remember this one. This one is nicknamed the guardian of the guardian angel of the genome, or the guardian of the genome. I remember me saying that in class. The p53 protein can turn on genes that are directly involved in DNA repair. If the DNA can't be repaired, the p53 gene activates what they call self-destruction genes, or you might remember hearing the word apoptosis this year. So the reason they call it the guardian of the genome is because it essentially is like the cell's self-destruct button. If this is not working properly, cells will not be destroyed in the event that they should not be dividing. So the top picture is showing you a normal situation, a normal scenario, where you have a it's normal in that this is what the protein is supposed to do. I apologize. So you have a mutagen. UV light, UV rays could be something that damages your DNA. And the DNA that gets damaged, that message is sent along this protein kinase cascade, which again is just like a domino series of messaging proteins that pass a message along to the active form of the p53 molecule, because this p53 gene is functional and it is working, which then promotes transcription of a protein that inhibits the cell cycle so that damaged DNA is not replicated. So the whole job of the p53 gene is to stop damaged DNA from going through the cell cycle. That's its whole job. That's what it's supposed to do. However, if that gene were to become mutated, it cannot activate transcription, and that damaged DNA then is going to be sent through the cell cycle. And now you have this mistake or this mutation being replicated over and over and over again in all these new cells that are being divided from the original cell. So it's supposed to stop that from happening. If this gene is mutated, you get damaged DNA, mutated DNA, replicating over and over again in your cells. They say that about half of all cancers relate to a mutated p53 gene. So the incidence of cancer generally increases with age. I've said that many times in class, generally speaking, right? Of course, there are cases where younger people get cancer. Of course, that's terrible, devastating, of course. But generally speaking, it's older people who tend to have a higher frequency of cancer. And this happens, and I've talked about this with you guys before, because mutations tend to accumulate throughout the course of your lifetime. So if mutations like in this picture here don't get caught and the cells don't get destroyed and they start to replicate, now you have more and more cells with this mutation, right? This damage. So your book uses colon cancer as an example. Colon cancer develops gradually. It starts with a polyp. So if you have older relatives, maybe your parents, they may have gone to have a colonoscopy done, a rather unpleasant experience, but they're looking for a little benign cell growth, or little benign almost tumors, they call them, or little growths called polyps. And they're looking to see if they're in there. Usually you might have one or two and they're removed. If you have a higher frequency of colon cancer in your family, there may be more, and you may have to go for colonoscopies more frequently. And they'll check those polyps to make sure that the cells are showing normal. A polyp is a result of cells that are dividing frequently, and that can grow and spread to other tissues, and that's when it becomes malignant. So the gradual accumulation of mutations that are converted proto-oncogenes to oncogenes, they knock out those tumor suppressor genes. Usually the RAS and the p53 gene are involved in these types of cancer. In order for a cell to become fully cancerous, there needs to be at least one active oncogene and mutations in several of the tumor suppressor genes. So here's a better sort of explanation of all that with a picture here. This is the multi-step model for the development of colon cancer. So first, you have the loss of the tumor suppressor gene that allows the polyp to grow. So here's the polyp in the picture here. So normally, if the tumor suppressor genes were working, it would have been like the brake pedal and slowed down this process of this little growth here, and it never would have occurred if this gene was fully functional. Next step here, the RAS oncogene becomes active and the tumor or the polyp starts to grow. So now you have out of control cell growth, if you will. It's growing larger. If the p53 gene is not functioning or it is off, mutations will start to accumulate in these cells until it becomes a malignant tumor. So you don't want to get to here. The whole point of going for the colonoscopies is to have them, hopefully it looks like this, right? But if you have polyps, and this isn't until you're, much older than you are now, or maybe they say it's around age 50, but you might go a little younger if you have a higher frequency of colon cancer in your family. But hopefully it's just something like this, one or two, and they're able to remove them and you come back more frequently, because once you have this situation happening, we talked about malignant tumors and how the cancer is going to spread to different parts of the body, different tissues, and then it becomes extremely difficult to treat. So your book also talks about breast cancer, and we've talked about that a little bit this year. There is a genetic component to cancer. People can inherit an oncogene or a mutant allele of a tumor suppressor gene, and there's strong evidence that 5 to 10% of breast cancer patients have inherited mutated versions of those genes involved in breast cancer. We've talked about these this year, the BRCA1 and the BRCA2 gene. Both these genes function in the cell's DNA damage repair pathway. They're kind of their own, almost kind of like their own specific p53 genes. They deal in preventing mutations from accumulating in breast cells, cancer cells. If that were to happen, you get a buildup of mutated DNA and essentially cancer. DNA breakage is the last thing that we'll talk about here in this chapter. This is the term, or this section, this is the term associated with mutations caused by mutagens. So mutagens, if you remember from Honors Bio and previous discussions, these are things in the environment, and there's a couple listed here: UV radiation, chemicals, something in your environment that are essentially cancer-causing agents. We also talked about this year how viruses can be responsible for different kinds of cancer. If you guys remember Henrietta Lacks and HPV, right, the human papillomavirus, that virus inserts its its genetic material into your cells and essentially takes over, incorporates itself into your genome, and that's going to interfere with gene regulation. You can get out of control cell growth from something like that, and we're going to cover viruses too, hopefully when we're back in school, but it may be remotely. All right, so that is section 3 of chapter 16. That's it. It's a short chapter. I posted a reading guide for this chapter. There's also a very quick mastering bio to do with this chapter, and by quick I mean it's going to take you less than 5 minutes to do. Just to review some of this stuff. A lot of it is review from chapter 9, but you have a couple new things here. All right, take care, everybody.